Today we're going to talk about cartilage and bone. Cartilage and bone is the skeletal connective tissue components of the body. We're going to talk about the general organization, the growth, extracellular matrix, and the different cells of cartilage and bone. Enjoy! Cartilage and bone. What we want to do is look at the general organization of cartilage and bone, the mechanism of growth, and characterize the different cells of cartilage and bone. If you look at connective tissue in general, you see there's a host of cells that are produced from stem cells in the bone marrow. All the white blood cells, fibroblasts, a host of cells are produced from there. But in addition to that, there's 5 cells that's associated with cartilage and bone. We have the chondroblast, which gives rise to chondrocytes. You have the osteoblasts, which give rise to osteocytes. And then we have the osteoclasts, which is derived from monocytes. It's a macrophage-like cell, multinucleated phagocytic cell. If we start with cartilage and bone, we see there's 3 basic types of cartilage. There's hyaline cartilage, which is what you mostly are familiar with, the end of a chicken bone, elastic cartilage, like it's in your ear, and there's fibrocartilage, which connects tendons and ligaments to bone. The early skeleton in our body is cartilage, and here we can see where the cartilage is replaced over time with bone, and we'll talk about that more later on. If you look at the ends of growing bones, you can see there will be a cartilage plate up there, and in there's different zones. There's zone of reserve, zone of proliferation or division, zone of hypertrophy when the cell's enlarged, and then you get the zone of calcification, and then finally the zone of erosion, where no longer do you have much cartilage associated with that. What happens is the cartilage is calcified, and when it's calcified, nutrients can't go through, the cells die, and as a consequence, osteoclasts move in to eat them, and it's remodeled at bone. So we have cartilage, bone, bone spicules, bone marrow here. This is where you would have compact bone on the either side there, skin out through there. If we look at those in one of the specimens, we can see the zone of reserve, zone of proliferation, zone of hypertrophy where the increase, zone of calcification, and then zone of erosion is what happens. Here we see the fetal finger. We look in this area right in through there, we see the cartilage and these spicules of bone. These are primary spongiosa because they still have a cartilage core. So they were all cartilage at one time, but the cartilage is being replaced by bone. Here we can see in the elbow the different layers of reserve, proliferation, hypertrophy, calcification, and erosion. As we can see there, these have been identified here: reserve, proliferation, hypertrophy, calcification, and zone of erosion. Here we see the tibia of a fetal tibia, showing you where the primary spongiosa, where you still have some cartilage core, but it's being replaced by this red bone. Bone marrow here, blue is the cartilage, and you can see the areas there of hypertrophy, for example. The typical cartilage that we see is hyaline cartilage, and we can see the hyaline cartilage here in the trachea. This little piece of trachea shows you the pseudostratified columnar epithelium with ciliated cells, but here's the cartilage here, and around the cartilage we have connective tissue core, the so-called perichondrium. If we look at cartilage with an electron microscope, we can see the perichondrium is type 1 collagen. There's fibroblasts out here, and we have collagen of type 1. In contrast, in the cartilage itself, so this is a capsule, perichondrium, this is a cartilage. you have chondrocytes trapped in their lacunae or little space, and we have type 2 collagen. Type 2 has a less distinct periodicity that we have in type 1. So this would be the capsule, and this would be the cartilage, and this is an example of appositional growth where you grow from the sides. So these cells, progenitor cells, like fibroblasts, we can't tell them apart, will be the chondroblasts, and then they will give rise to chondrocytes, which are trapped in their lacunae. Another type of growth a cartilage has is interstitial growth. That is, it can grow from within, and one cell can divide to make two, and you can see where they kind of pushed apart one another, illustrating the interstitial growth as opposed to the appositional growth which occurs on the side. Cartilage, the hyaline cartilage at the end, is also involved in the growth of long bone. The epiphyseal plate is cartilage in through there, which will grow by proliferation and then is transformed into bone. Bone cannot grow within because the minerals prevent you from stretching bone. So the cartilage can grow from within. It grows from within, then it replaces bone, and that's how you grow your long bones in your body. And we call that is endochondral bone formation, where you have bones formed from where cartilage was at one time. And here we can see some bone cells. This is osteoclast, a multinucleated phagocytic cell that's eating bone and also is eating cartilage in through there. We also see osteoblasts. Osteoblasts are on the edge of the cartilage, and in the very center there we see osteocytes that get trapped in their lacunae. Again, we see some osteoclasts, osteoblasts on the sides, laying down more bone. Here we see some more osteoblasts, which will give rise to the bone. Now, another type of cartilage is elastic cartilage, and that's what we see here. We see the perichondrium that goes around here, connective tissue bound around the cartilage. It's very similar to hyaline cartilage, except it has a lot of elastic tissue in there. And one of the places you find that is in the outer ear. Here you can see skin from the outer ear here and here. Some fat cells, also connective tissue. But the purpose of this is elastic cartilage, to be able to reform to the original shape. You can bend your ear and it'll pop right back to the original shape that it was. So elastic cartilage is what we see, and connective tissue bands on either side, which are the perichondrium, epithelium on the outside. The third type of cartilage is fibrocartilage, and that is something that's intermediate between a ligament and bone. And so what you have, instead of having fibroblasts coming down, so you have chondrocytes located in here, and so these cells differentiate into chondrocytes, and then it hooks onto the bone. So where a tendon or ligament might hook onto the bone, we have the intermediate cartilage in between there, and that's a fibrocartilage. And we can see the fibrocartilage here. So this is type 1 collagen running through there, which is typical of a tendon or ligament. And then here we can see the fibrocartilage cells within there. A higher mag of that, these are the chondrocytes surrounded by the connective tissue of the tendon. Again, so these are type 1 collagen in through there, and chondrocytes in this fibrocartilage. Here's a typical tendon, which have just regular fibroblasts with lots of type 1 collagen, as we see there. In contrast, the fibrocartilage that we see there has chondrocytes in there, which has a little lacunae, a little space that you see in there, unlike what you see in a typical fibroblast. So endochondral bone formation is when you start with a cartilage, and that's remodeled by bone. And what happens is the cartilage becomes calcified. No longer can nutrients diffuse through. It kills the cells, and osteoclasts eat it up, and then it's replaced with bone. If it's on the side through here, it will be replaced by compact bone. If it's in the middle, it'll be replaced by spongy bone or cancerous bone. Compact bone, cancerous bone is what we have. Now, if we look more closely at the cells, the osteoblasts, they secrete osteoid. Osteoid is bone material, and that's what we see right here. So the osteoblasts along the edge secrete that. Expands the bone by appositional growth. So as you have another layer, it expands the bone in this direction, that expands in that direction. It does not grow from within. It's only from the side. Osteocytes are osteoblasts that are trapped in their lacunae. So whenever this has a matrix around this osteoblast, it becomes the osteocyte, as you can see there. Now, bone grows by appositional growth. You got differentiation of cells that give rise to osteoblasts, which then gives rise to osteoclasts whenever they're trapped in their little space or lacunae. One of the important bone cells is the osteoclast, and the osteoclast has a ruffled border, as you see there. It's not ruffled everywhere, but it's really ruffled here, and in this ruffled border facilitates having a very low pH and has a lot of citrate in there, which is able to break down the hard components, the mineral components of bone. And breaking down the bone is very important in maintaining calcium levels. The parathyroid hormone causes bone reabsorption. Calcitonin prevents reabsorption from occurring. These hormones tightly regulate the amount of calcium. 1/4 of free calcium in the blood is exchanged every minute. So there's a lot goes on. Also, bone functions in hematopoiesis. That is, it provides a cavity for the blood cells, white blood cells and red blood cells to go. Now, compact bone on the side has these Haversian systems, that is, concentric layers of cells that have developed and laid down with a matrix materials around it. And so each little cell is a lacunae, is where it is, and it's attached to other cells by these little canaliculi, and that's how it gets nutrients from one to another. So you have a major center coming through there. That is your Haversian canal, and then the ones on the side are the Volkmann's canals that bring nutrients to this. So this is one unit of growth, another unit of growth. This is one unit of growth with all these little spider legs, which are the canaliculi, which connect the two. Now, when bone is remodeling, what happens is the the osteoclasts, they make the hole, and then the osteoblast just covers up the hole. As you can see, it's laying down here, and then a previous layer was laid down here, and a previous layer was laid down there. So eventually you're going to close this up, and since this is a circle in through there, it's going to make could make compact bone. And here we can see what happens with compact bone. You get osteoclasts have like a forming reabsorption cavity. That is where they're eating a hole. And these are the cells that are directed. The electrical charge in the when your crystals are pushed together direct these cells so they go down the length of the bone as opposed to out the side, and that makes a hole. and then this hole, so you have reabsorption cavity. You got a cavity here now. So this is a cross section of this, and then the osteoblasts will lay down more bone. And here you can see where one group is laid down, another group is laid down, another group is laid down, and eventually you close in the whole Haversian system. Partly closed here, completely closed there. Nutrients in the center, but you have these different rings. As you see, this is compact bone remodeling, and this happens over time. You dig a hole and then you fill it back up, and the osteoclast that makes the hole is what directs the process. And here you can see different generations of remodeling bone. This is one Haversian system, and then the second generation is the osteoclast eats up another hole, and sometimes it can be the previous one. Here's the third generation. You can see 1, 2, 3 generations right there. It didn't eat up everything. It just eats up at random, sometimes the interstitium, sometimes the previous one, but this is how it happens when it remodels. Why would you want to remodel? You remodel bone to adjust to stress. If you start lifting weights, you need heavier bones, and so you remodel to make the bones more heavy. This is a problem living out of space, is you don't have weight, and as a consequence, the bone remodels for lightweight, not heavyweight. So we can see the compact bone, the Volkmann's canal, the Haversian canal, and we can see how the collagen fibers are laid down perpendicular to one another, as you can see, as they make these concentric rings, and they fill up from the outside in. So these are the youngest ones. It's just opposite of a tree. The youngest is on the inside, not the outside, in these Haversian systems. Here we see a Haversian system with the osteocytes located in the lacunae and the canaliculi projecting from one to the other. This is a source of nutrients in the Volkmann's canal, and then it migrates from this cell to that cell to that cell, and it passes where the cells touch one another and have gap junctions between the two. Here again we can see the Haversian canal. This is a real specimen here with the little canaliculi, which fingers from one cell touch another cell, and the gap junctions therein facilitates nutrients from moving from the center of Volkmann's canal out to the outside. Again, Haversian system here. There's another Haversian system in through there, and we can see the different layers. So this developed from out here, and then this group, then that group, this group, as it grew by appositional growth. So it didn't grow from within, it grew by more cells laying down on top of it. And actually that is not the periosteum, that's the enosteum on the inside. So outside the bone we have periosteum, on the inside we have enosteum. And we see a lot of fat cells and bone marrow in this particular one. But if you look at the compact bone, you can see the Haversian systems, how this is a primitive form of those. Those will be remodeled and be more organized than what you have. But this is a cross section of this bone, and the Haversian system run down the length of the bone. Again, we can see the Haversian system, also called osteon, but it has these osteocytes trapped in their lacunae, is what we see. And so Volkmann's canal, this is a Volkmann's canal right here. I don't see sorry, the Volkmann's canal is the one that runs perpendicular. This is the Haversian canal that we see there. This is a tibia. Shows you again where cartilage is being remodeled on this compact bone. There's a bone marrow there. So this is compact bone that we're looking at right in through there, and you can see it was cartilage at one time. It's being replaced by bone. Here we see some osteoblasts laying down more bone in this particular specimen. We see some osteoclasts in through there breaking down bone. These are osteoclasts, and you always see have a constant breaking down and remodeling of bone to adjust to whatever's the environment, and also to adjust to the calcium levels. Osteoclasts, osteocytes, osteoblasts. So these become sites whenever they get trapped in their lacunae. Also, if you look inside the bone, you'll see bone marrow. Here we see some eosinophils. There's a host of different cells, a lot of red blood cells in through there, and a host of cells that ultimately will be immature cells will make red blood cells and white blood cells. Here we can say, how many osteoclasts can you find? Osteoclasts, there they are. There's one up here, there's one down here, there's one right there, there's one right there. These are multinucleated phagocytic cells that break down cartilage and bone. So we have endochondral bone formation. That is, you start with a cartilage and that's replaced by bone. You have a primary center of ossification, which would be the primary center. You also have a secondary center, and this is a secondary center, the apophysis. Diaphysis, the apophysis, is the secondary center. The more complex the bone is, like say a bone in the vertebrae, very complex with the projections, that has more centers of ossification to occur. Cartilage growth by endochondral bone formation. Cartilage grows replaced by bone. Here we see where cartilage forms the articular surface with other bone, and that's a good advantage of cartilage, is it able to support weight and also slip and slide. Now, in another type of bone formation is intermembranous, and that is it doesn't use cartilage. What it does is it goes from fibroblastic-like cells, progenitor cells, osteoblasts, and give rise to osteocytes. And here we can see some of that occur. Flat bones of the skull. Here you can see a little embryo developing the cells in the skull. So basically we're talking about the skeletal connective tissue of the body. Let's go over a few questions from the midterm didactic exam. It was taken in 2012. You're supposed to choose the best answer for 2 points each. Which item is or characteristic of epithelium? Secretory cells of glands? Yes. Covers organs? Yes. Line lymph vessels? Yes. So the answer is E. Second question, connective tissue is characterized by being loose or dense connective tissue? Yes. Cells usually sit on a basement membrane? No. Avascular generally? No. Only cartilage is avascular, so the answer is A. Muscle have microfilaments and myosin arranged diffusely or in sarcomeres? Yes, diffusely in smooth muscle, sarcomeres in striated muscle. Different functions of skeletal, cardiac, and smooth muscle? Yes. Low density of contractile elements? No, they have a high density of contractile elements, so the answer is D, A, and B. Nervous tissue may be composed of cell bodies outside the brain and spinal cord? Yes. Composed of myelinated and unmyelinated nerve fibers? Yes. Neurons are large excitable cells with long processes? True. The answer is E, all the above. Which vessel characteristic match? Capillary, narrow tube, lined by single endothelial cells? Yes. Vein, large irregular shaped lumen to wall thickness? True. Lymphatic, small, thin-walled, carries blood? No, it carries lymph. So the answer is D. So today we talked about the cartilage and bone, which is a skeletal connective tissue components of the body. We talk about the general organization, the growth, extracellular matrix, and the cells of cartilage and bone. Thank you.